You know, in order to understand that, it's important to do a brief history lesson on how discovery actually happens in the laboratories. You know, many, many years ago, decades ago, the standard of scientific discovery was really the test tube and slips underneath a microscope. And that changed because there was really a need to do higher volumes of screening. And entered a media called the microtiter plate. And if you can see this, essentially a microtiter plate has 384 microliter level test tubes embedded in it. And what evolved over that time was a whole industry of automation to support this particular standard of media in the laboratories. That automation looks like robotics, automated and motion control devices that actually would move this particular microtiter plate through the laboratory process. But what we're seeing right now is that this particular microtiter plate media has actually become an inhibitor. Given the increase of screening demand that's being projected by scientific laboratories, this will need to change. The cost per data point also associated with this is prohibitive from an automation standpoint as well. What's required? A shift, a disruptive paradigm shift. And enter Douglas Scientific. We have a patented media called array tape. And if you can imagine each one of those microtiter plates now being reduced down in size to nanoliter level volume in embossed wells on a continuous array tape platform, polypropylene, polycarbonate, polystyrene, or maybe any other polymer, we've actually devised a method and a media to conduct laboratory process at a much higher rate. This inline polypropylene tape has embossed wells, and each of these embossed wells has a very unique geometry to the scientific application that's done in the laboratories. It's flexible and low cost to be able to accomplish this. As I mentioned, Douglas Scientific also has developed a full line of instrumentation to support this array tape continuous platform. What you're seeing right here is something we call the Nexar, and you can see the reels of tape, which just literally have meters upon meters of those assays being managed. This is an inline liquid handling and sample processing unit. We also have engineered and deployed a full line of scanners. This is called the Arrea, that does inline fluorescent detection. It excites the experiment within those wells and then captures the data and takes it out to full and sophisticated LIMS systems to do the data analysis. Well, let's take a brief look at how that works. So this is our system in a very brief video segment. You can see the Nexar is actually working with tape, and here 384 pipettes are aspirating DNA compound, taking it back into the Nexar, dispensing it into those 384 wells on the tape. What you're going to see here is now another dispense head further down in line that's actually adding chemistries. Those might be markers, those might be compounds that are actually conducting the experiment in those wells. Everything's barcoded and captured on a per well basis. And here you can see that the tape is being sealed for offline processes such as incubation and PCR that may be done in the labs. This is the scanner. You can see the fluorescent lights are actually exciting what's in those wells, identifying markers, capturing the data, and importing and exporting it out to LIMS systems.